So today it's my pleasure to welcome Amy Arnsten to the podcast. She's the Albert Kemp Professor of Neuroscience at Yale University, where she's also a member of the Kavli Neuroscience Institute. She's an expert on how the brain works and what goes wrong in both psychiatric disorders and Alzheimer's disease. She's had a lot of focus on the prefrontal cortex, which, if any of the viewers and listeners of this podcast have looked at some of the previous podcasts, we've had several on the prefrontal cortex and its roles in decision making and the evolution of the prefrontal cortex, because it's the most highly evolved region of the brain. So Amy, you want to start out by giving our listeners and viewers some background on you. Where'd you grow up? How did you get interested in the brain?
Sure, Mark, and thank you so much for including me. So I grew up in Maplewood, New Jersey, and I was a Unitarian. My parents took me to the nearby Unitarian church, which was also attended, I think half the congregation were scientists from Bell Labs. My Sunday school was all about science and the wonders of nature. So I was immersed in that, create the future in an optimistic, wondrous way, right from early childhood.
That's interesting you mentioned Bell Labs, because and you'll talk a little bit about calcium, and we've had some common interest in the role of calcium in normal function of neural circuits and in disease. And so when I started out looking at calcium, it was shortly after Roger Chen had developed the calcium imaging dyes like FIRA2. Actually, John Connor at Bell Labs, I knew him really well, and we actually published on one review article together. But for those who don't know, Bell Labs used to have a really active, top-notch science and neuroscience research program, basic research.
Yeah. So that, when I say they created the future, including they created your research. Yes.
Yeah.
They were amazing. And it was before, this was in the 1960s. So moonshot and a very optimistic time in science. So wonderful to hear that they even influenced your work.
Yeah.
It was a privilege to be part of that. Yes.
And then you went to Brown University.
Yes, but before that, in terms of motivations, when I was a teenager, my father had a major heart attack, and I watched as the whole medical community knew just what to do, saved his life, tremendous outpouring of compassion towards us. And about the same time, his sister had what they would call a nervous breakdown. And it was the exact opposite. No one knew what was happening, what to do, the stigma, hide it away. And I was so struck that for a heart, we had all these rational therapies. For brain disorders, we knew nothing and we were hiding away, and that there was just this critical need to understand the brain, to be able to treat brain disorders the way we were treating heart disorders. So that really motivated me tremendously for the whole direction of my research. And yeah, I went to Brown, and I chose Brown in part because, and it still has this, you can create your own major. It's a very, a place where you can not only be creative, but take responsibility for your future. I planned what courses. I knew I needed lots of organic chemistry. So I enjoyed it because I was assigning it. And it turns out in so doing, I became the first neuroscience major. So I created the neuroscience major at Brown, and they've done a great job with that ever since. and then heard about the locus ceruleus at Hampshire College, because I took my junior year abroad in Massachusetts at Hampshire, because they actually had great neuroscience at that time, when most other places did not yet have it. And I learned about the locus ceruleus and how these noradrenergic neurons went throughout most of the brain. And I became very intrigued. And so went to UCSD for grad school, because there was a lot of research on locus ceruleus there, and did my thesis on that. But while there, this woman came to give a seminar, and that in itself was rare back then. And her name was Patricia Goldman-Rakic. No one had heard of her. It was a tiny seminar room with like 10 of us in attendance, and she talked about the prefrontal cortex. I realized this is what I had to study if I was going to understand what goes wrong in mental disorders. So I changed my whole life to come and work with her, and I've been at Yale ever since.
Wow. And yeah, so she is a pioneer in studies of the prefrontal cortex. Could you kind of briefly, I mean, her career is well storied and documented. She, not only in her publications, but in her honors and awards, she was a member of the National Academy of Sciences. She was president of the Society for Neuroscience, which, you know, is, as you know, when you go to a neuroscience meeting, there's 30,000 plus people there. So it's a big field, and she was a pioneering woman for sure in the field. Yeah, she did a lot of work, but it was background for years, and you worked with for your work. And so it's good for you to kind of summarize what her major findings were, and then how you have been taking that a step further, getting down to the molecular and cellular level.
Yeah, so I don't think people these days can understand how little was known back then. Everybody was studying the primary visual cortex, and many scientists would say that you couldn't use scientific methods to study prefrontal cortex. It was just this kind of mushy area under the forehead. It was the place where neurosurgeons were happy to take tissue out because it wouldn't have an effect. It was a very sensory motor time, and things like thought were not thought to be scientifically approachable. Yeah. And so she, not only as a woman, but as someone who could knew how important this was, really tackled it, which is amazing. I might say that I think when you're an outsider, and I have felt an outsider as a woman, you know, you're often the only woman on the hallway, in the room. Sadly, that sometimes still happens. It helps you to think outside the box, right? You're left alone to really dwell with the data, and they can bring you places that other people's thoughts and the habits that have developed in a community can be a trap. And so there can be advantages to being an outsider. And she thought magnificently about prefrontal, and one of her great clues that it was going to be possible to solve it was she had gone to work with Wally Nauta in MIT to learn how to do anatomical tracing. At that time, she was at NIMH, and she was doing wonderful work as a developmental neurobiologist, but she wanted to learn track tracing. Her husband, Pashka, would say it's because he was in Boston and she wanted to be close to him. So that probably figured in, but it had great after effects, because she learned these new methods for sensitive anatomical tracing. And what she saw in prefrontal cortex were columns, just like there were columns in primary visual cortex. And to her, that was a talisman that, okay, that meant this area of cortex was also organized. It would also be amenable to scientific approach. And from then on, she went at it using whatever method she needed to ask the question that was most important. And that too was really different then. Back then, you did a method and you stayed in your lane. She, with her prefrontal cortex, did this top-down instead. The method's irrelevant. It's the question that matters. And so she would bring people into her lab group who were experts technically to address a question. And with this, so Jochen Fuster had just discovered that neurons in prefrontal cortex are able to keep firing without sensory stimulation to keep information in mind, persistent firing over a delay period in working memory. He was using the methods developed actually at Yale in the 1930s, working memory tasks, which were manual, where the monkey would watch you hide a treat. There'd be a screen that would come down for a delay period. It would raise, and then the monkey had to use its memory of where that treat had been hidden to get the reward. And the position would change over dozens of trials, so constantly updating which well was it in this time. And back in the 1930s, Jacobson had shown that lesions to dorsolateral part of prefrontal cortex produce massive and permanent deficits on that task. And he said it was as if out of sight, out of mind were literally applicable. That these monkeys could solve all sorts of puzzles and tasks as long as the information was available in the environment. As soon as they had to represent it in their minds, in working memory for even a few seconds, chance performance.
That's interesting. I just had a thought. So I've been playing a lot of chess online, and so the chessboard is always there, but then you can actually close your eyes and kind of visualize and keep in your memory the positions of the players, and even kind of do the chess game in your mind. So I imagine that's the same circuits that are involved in that.
Exactly. Dorsolateral prefrontal has very strong reciprocal connections with the parietal association cortex that does visual spatial position. And just for fun, to add, my husband's a big chess player, and we had a chess club when our kids were little, and he would impress them and thus keep them in line by playing them all blindfolded. So it was, for him, it was
wow,
his memory. Yes.
He's better than I don't think I could do that.
We'll see. We'll give you some meds to increase your prefrontal.
Yeah, maybe we can connect on chess.com and.
Yes. Anyway,
okay. These lesion studies in the monkeys, there was that kind of background information before Pat started, and then she wanted to really understand what are the connections between the neurons. So she's tracing the connections, between different neurons.
Yeah, so the first thing she did actually when she was at NIH was discovered the sub area in prefrontal that was essential for visual space. And so then she looked at the anatomical connections of just that area. And indeed, it was with this part of parietal that does visual space. Yeah. Also with hippocampus for being able to partner for longer term memory, because you need hippocampus if the delays are longer than like 15 seconds. Connections down to the striatum for and to motor cortices for influencing actual motor response. Connections with emotional areas like the orbital prefrontal, areas that are specialized for evaluating reward value. So, and then she also saw that just below that, there were circuits that instead of connecting with areas that were specialized for visual space, it was areas that are specialized for visual features. So the temporal lobe, the ventral stream. And then Leslie Ungerleider at NIH went and showed the same thing in using human fMRI, so that these parallel circuits that Leslie's group had been involved with tracing from primary visual coming out, that these parallels continued all the way up into prefrontal, of course, with these areas extensively talking to each other. And then Pat was able to do physiology, and with the anatomy, she was an exquisite fine motor control. So she did these tiny little tracer injections and was able to see these hugely extensive recurrent excitatory circuits. So connections horizontally, specifically in deep layer 3 of dorsolateral prefrontal. And this is the layer that just massively expands with brain evolution. It is the layer that is, or sublayer, that is most affected in schizophrenia with spine loss, dendritic loss. These are the cells most affected in Alzheimer's disease that develop tau pathology and autophagic degeneration. So bullseye for these circuits, that what she then showed, the physiology fit with this, the anatomy, that these cells are able to excite each other to keep information in mind without having to have bottom-up stimulation. And that this extensive glutamatergic recurrent excitation is part of what makes these circuits so vulnerable.
And then you started digging deeper into what's going on in those layer 3 pyramidal neurons in terms of, what neurotransmitters, what receptors, and so on. So I think
yes, she began that work. I mean, that's why she brought me in as her postdoc. We're supposed to look at dopamine and aging, and I was doing norepinephrine as sort of a control. And I should say she did this pioneering work back in the 1970s while still at NIH, doing catecholamine depletion of dorsolateral prefrontal cortex at a time when the catecholamines really were just being discovered as being part of brain. So it was so early on. And that work showed that if you deplete prefrontal of catecholamines, it's as if you've taken out the cortex itself, that these were essential. So that was a major clue. And that was part of why I went to work with her. And so everybody at that point was in love with dopamine. It was the brand new catecholamine, norepinephrine, old hat. And I looked at the footnotes in their data and I saw, I think it's actually norepinephrine is really doing much more than you think. And I looked at their data, and sure enough, the pattern really fit with norepinephrine also having an important role. And that's why if you get rid of both of them, you're in such trouble. So that work led to discovering that norepinephrine has this very important beneficial action at alpha-2 receptors, specifically alpha-2A adrenoceptors. Alpha-2 receptors have really high affinity for norepinephrine, so it doesn't take too much to engage. And what we've now shown is these beneficial effects occur with moderate norepinephrine release when we feel safe, interested, alert. But when you are stressed, you get really high levels of norepinephrine release. Those engage low affinity noradrenergic receptors, alpha 1 and beta, and those impair prefrontal function and take prefrontal offline during stress. So norepinephrine is a really powerful switch for which brain systems control our behavior and thoughts, because amygdala, for example, is the exact opposite of prefrontal. It's weakened by alpha 2. Boring, we're safe, who cares, nobody needs an amygdala, and strengthened by alpha 1 and especially beta 1. So we're under stress. This is amygdala's time to shine. There, so amygdala becomes in control under threat, and prefrontal is shut off. So I think it's a really cool.
And that makes sense from an evolutionary perspective, because the animals in the wild, and presumably our human ancestors, we had stresses in our life, but they weren't chronic. They were kind of temporary, and it's like, just get away from some danger. And so then maybe it's okay to shut down the prefrontal cortex in that situation, get away from the danger, and then the prefrontal cortex comes back online, and you can do some critical thinking.
Yes, I think the equivalent in our life is like you're cut off on the highway. There you are thinking about, well, I have to do this and this and this, and maybe I should have it like this. And boom, here comes this SUV right in front of you. You stop thinking, you just slam on the brakes, and amygdala can activate striatum, that habit to slam on the brakes. I do think, though, that a lot, when we speculate about these evolutionary mechanisms, and it's of course so fun because there's no way to prove us wrong, that the way people have often thought about it, acute stress good, chronic stress bad, but you can see where these mechanisms probably were really important for chronic stress too, like long periods of starvation, periods where there'd be continuous threat outside of the burrow or cave. And some people think this is why we have depression, that it was an evolutionary advantage. You hibernate. So this kind of, it's not just fight or flight. There's also freeze, and this kind of hiding away that happens with chronic stress. You can imagine in a sustained threatening environment that had survival value as well. But of course, what we're pointing out here is that there are threats where you need prefrontal to survive. A whole society needs prefrontal to survive. And we've just been living through such an instance. Viruses that are invisible. The amygdala has no concrete clue that this is a threat. You have to generate this abstraction that this is a threat, and so you need prefrontal. I should probably, we're just assuming people know what prefrontal does, so why don't I quickly, if they've listened to your other podcasts, they'd know, but in case not, you're probably already getting the sense that it's so important for representing things when they're not actually happening. So having those recurrent circuits generating something without any sensory stimulation. And of course, this is what we call working memory, but it's also cardinal to abstract thinking and being able to dissect things and then think about them logically. So higher reasoning. And it's critical for guiding thoughtful behavior, thinking into the future, near future or far future. Will this be good for me? I know it might seem really wonderful, delicious to do now, but I'm going to pay for it later. So these are the executive functions: top-down control. No, let's not speak out. We're going we're really angry right now. You want to say this thing, but this is our boss. We will not do well if we speak out like that. Speaking of speaking in humans, prefrontal is key for language production. This makes sense. Language is symbols, things that aren't in the environment. They're internal, and most people, their language generation is on the left side, and emotional coloring of language is on the right. For left-handers, that can be reversed or mixed. So that's something we can't study in animals, but important to know, especially there's evidence of laterality emotionally too in humans, where the left hemisphere is the cheerleader, hope, optimism, you can do it, keep going. And the right is the no, don't do this. This is what's going to cause trouble. And so you really see that in disorders like bipolar disorder, where when you have loss of activity in right hemisphere, you have mania. So someone disinhibited. When you have altered activity in the left, you have depression. And this is why what's called rapid or repeated transcranial magnetic stimulation, the rapid form of it to strengthen, is put over the left dorsolateral prefrontal to help with depression, whereas they try to weaken the right side with depression. So that seems to be something in humans, this laterality, that's really important. Other things prefrontal does, so that you're probably already getting a flavor for: planning and organizing for the future, including near future, like the end of a sentence, in a very profound ways. Flexible decision making, which your podcasts have talked about, with really sophisticated evaluation of rewards, especially rewards that might happen in the future that aren't even there right now. And the frontal pole, which expands tremendously in human for metacognition, thinking about thinking. I have to remember to remember, things like that. And also theory of mind, trying to figure out what somebody else is thinking. Are they trying to fool me? And that can be really important for what we're seeing now with misinformation. That there's evidence that people with lesions here can be fooled by misleading ads. And I would say misleading political information, same thing. And the reason that's also so important is this part of prefrontal is tremendously vulnerable to stress. We lose gray matter there with chronic stress. So someone chronic stressed is now going to be more vulnerable to misinformation. So also insight and judgment in these areas, and empathy and moral conscience. So you see just so much of what makes us human.
Everything that makes us human is going on. And that's great. So let's go back to these layer 3 neurons, pyramidal neurons. They use the neurotransmitter glutamate, and then you have inputs from these neurons that use norepinephrine or dopamine. So glutamate activates receptors. There's membrane depolarization. Then you get influx of calcium through channels, in particular what's called the NMDA receptor channel, which is actually a glutamate receptor. So glutamate binds. The membrane depolarizes, and then calcium goes through into the cell. So you've got calcium going in response to glutamate. And then what's happening in response to activation of the norepinephrine or dopamine receptors?
Well, first of all, let me say a little bit more about that NMDA, because it's really important.
Okay.
In a typical glutamate synapse, you have not only NMDA, but AMPA receptors. Most AMPA receptors don't flux calcium, but what they do is depolarize the synaptic membrane. So now the NMDA receptor ejects its magnesium and can respond to glutamate, and we can have sodium and calcium entry. Prefrontal, or at least these layer 3 circuits, seem to have remarkably little AMPA receptors. When you block AMPA receptors, it's surprising how little happens. And what instead seems to be doing this important, what we call permissive role, depolarizing the membrane, is nicotinic alpha-7 receptors, which are right there in the glutamate synapse. And the reason that's so fascinating, acetylcholine, which stimulates these receptors, is only released when we're awake, not when we're in deep sleep. So we think this is part of why we're conscious when we're awake, but unconscious in deep sleep when there's no acetylcholine. By the way, acetylcholine is released in REM sleep. So is this part of why we are sort of aware during dreaming? So really fun questions, but also really relevant to calcium and its important role, because calcium depolarizes the synaptic membrane. And we think that these circuits, since they're not having bottom-up stimulation, have to have a lot of calcium to keep that synaptic membrane depolarized so NMDA receptors can work. And so what we've been finding is that prefrontal neurons express the molecular machinery to magnify calcium right next to that glutamate synapse. They have NMDA receptors that flux the highest levels of calcium, so the gluN2B. They have voltage-gated calcium channels right there, a lot of them. And they have the smooth endoplasmic reticulum, which stores calcium, comes into these spines and elaborates next to the synapse. And so we have internal calcium release. And all of these things interact, where, for example, calcium coming through the voltage-gated calcium channel causes calcium-mediated calcium release from the smooth ER, so that you can really have magnification. And what else we see there doing the magnification is cyclic AMP signaling. So cyclic AMP activates protein kinase A, which can phosphorylate ryanodine and IP3 receptors to increase calcium release from the ER, also increase calcium release or calcium entry through voltage-gated calcium channels and through the NMDA receptors. Then calcium in turn creates more cyclic AMP. And so you have feed-forward signaling, which can then blow up a lot of calcium. And so norepinephrine and dopamine can drive this through D1, alpha-1, beta-1 receptors. So alpha-1 through IP3, stimulation of IP3 receptors, beta-1 in particular through Cav1.2 calcium channels, and D1 through magnification of cyclic AMP. So we get to see that all of that's there to really blow it up. And what we also see there that's somewhat unique and really important and gets back to the stress response we were talking about and why prefrontal goes offline with stress, we see a collection of potassium channels that are opened by cyclic AMP, pKa, or calcium. And they're right there on those spines. And when they open, the synapse disconnects. And so it's a way, without any architectural change, you can take a synapse and basically have it disappear. Because now this neuron's trying to excite this one, but the potassium channels are all open. So it's like the information just leaks out before it gets to the dendrite. So this is a way you can instantly take prefrontal offline. The SUVs cut you off. Boom, stop planning your grant and just slam on the brakes. So it's fantastic, but now we have something built in to weaken prefrontal. And we get to see that if we don't tightly regulate cyclic AMP calcium signaling, we end up with prefrontal deficits. And with chronic stress, and calcium overload through mitochondria, actual loss of the spines and dendrites. So tremendous vulnerability.
And that happens in schizophrenia. Is that correct?
Yes, and with chronic stress. And those are very much interrelated with risk. The largest risk factor genetically for schizophrenia is complement C4a, which is one of the sequence of signaling proteins that signals microglia to eat the spine, for example. And we can see that when you get dysregulated cyclic AMP calcium signaling, you get activation of complement pathways. So the inflammation, aging, we see loss of regulation. So one of the main molecules that keeps this under control is phosphodiesterases. PD4 subtype A and D are right next to the smooth endoplasmic reticulum. They are gone with age or with inflammation. Another really important one is calbindin, and just a subset of pyramidal cells express calbindin. It's a calcium binding protein. So when you get a lot of that calcium out there in the cytosol, it binds it, keeps it under control, and we lose calbindin with age. And so then we get too much cyclic AMP, too much calcium, and end up with loss of firing, loss of synapses, and the driving of tau phosphorylation, which leads to tangles and degeneration. So we're able to see the beginnings of all of this in the very old monkey brains.
On Friday of this week, I'm going to do a podcast with Anne McKee.
Traumatic brain
CTE, the chronic traumatic encephalopathy. And has anyone looked at prefrontal cortex function in some of these, say, NFL players who are having problems? And then post-mortem, has anyone looked at these layer 3?
Yes. So exactly. No, it's a beautiful connection. Absolutely. Prefrontal is one of the most susceptible to tau pathology and degeneration and CTE. And what you see, so we think with normal aging process, it's happening slowly.
Yeah.
And if you live long enough and have enough risk factors, it turns into Alzheimer's disease. But with traumatic brain injury, it's happening. It's a large trauma and happens fast. And of course, traumatic brain injury is a major risk factor for Alzheimer's disease. But so is emotional distress. So studies out of Scandinavia and in California, where they have health records on people for decades, people who have trauma in their 50s, like loss of a child, have a much higher risk of having Alzheimer's disease 20 years later. So the fact that physical stressors like traumatic brain injury and an emotional stress can produce the same pathology, because when you get down to it at the cellular level, they're activating many of the same pathways.
Yeah, very interesting. Now, you want to talk a little bit more about Alzheimer's disease and what you're doing now? You mentioned before we started the podcast, you're also looking at a region of the brain that's affected very early in Alzheimer's disease, the neuronal cortex. And you said, in our conversation before the podcast, that there's some similarities in the neurons that get the tau and degenerate in the neuronal cortex and these layer 3 neurons.
Yes. So this signature of what we call flexibility, vulnerability, so having the magnification of calcium and then these potassium channels that allow this for very dynamic strengthen, weaken network connections. we're seeing an entorhinal cortex as well. And this fits with some of the rodent studies of entorhinal cortex, which are the grid cells.
Yeah.
And what they see is this dynamic change in grid scale based on arousal state, which is very similar to what we see in prefrontal, where we can see the representations of visual space. For example, with dopamine D1, they get to be small and very precise. And then you add stressful levels of dopamine and they actually disappear. You do other things and they can broaden. And that same kind of dynamic change in representations has been seen with grid cells. And we're seeing now, yes, indeed, they have the same signature of flexibility, which then, if dysregulated, it really confers vulnerability. And what's fascinating in humans, and we think probably in monkeys as well, this is a study we're doing now, it's the layer 2 cell islands in entorhinal that are so vulnerable to tau pathology. You can see pathology there, even in young adults, certainly in middle age, in humans, and also in monkeys. And these cells never have calbindin. So they're magnifying calcium to be able to have these dynamic changes in network connectivity, but they don't have this key protective factor. So we think that might be why it starts so early there. And then if you look at who has calbindin, the highest levels are actually in GABAergic interneurons, which don't die in Alzheimer's, and in Purkinje cells in cerebellum, which don't die in Alzheimer's or get tau pathology.
And also dentate granule neurons in the hippocampus.
Yes, and they are more resistant than the CA fields.
Yeah.
So this idea that, and then if you look across the sensory streams, V1 has very little, and then you get more and more as you go up into the association cortices. So we think that cells that have the most calbindin, or glutamatergic cells with the most calbindin, are needing it because they have magnified calcium signaling. But then when you lose calbindin, for example, with age or inflammation, such as you lose it with COVID, in severe COVID infection, now those cells are very vulnerable because they have magnified calcium without this key protector.
So someone has found that in people who died from COVID, there's a reduction in calbindin levels?
Yes, and there's tau pathology, including,
Wow,
and this is a huge clue, Mark, in cerebellum. So unlike in Alzheimer's disease, where calbindin remains in cerebellum, with severe COVID infection, so people who died from it, calbindin is lost from the Purkinje cells in cerebellum, and there's tau pathology in cerebellum, which there normally isn't. So it's a real link, I think, between these, that when you lose calbindin, you're far more vulnerable to tau pathology.
And of course, thank goodness a lot of people, survive COVID, but then the question becomes, there is evidence emerging that some people who've had COVID, maybe particularly those who had to be hospitalized, have some long-lasting abnormalities, including with cognition, I think.
Absolutely. And so the compound that we discovered that helps to strengthen prefrontal is now being used off-label. We're needing placebo-controlled trials, but is being used to treat cognitive deficits in long COVID. So it's called guanfacine. It's an alpha-2A agonist, and it's what I discovered as a postdoc with Pat, because I was then testing aged monkeys with these naturally occurring prefrontal deficits, which we then went on to see was related to too much cyclic AMP and calcium. And I was a postdoc, and one day testing this aged female monkey who was completely distracted, just such a ditz. And when we test, we're blind to drug treatment. So I was in there with her, and she was completely changed this one day. Instead of being distracted and all over the place and making errors constantly, she was like a laser beam. And I was like, what has happened? And we always have index cards that would tell us what had happened in case a monkey got sick, and we could call the vets right away and say, that actually never happened because we're testing things that improve cognition. So we never had to do it. But I broke the blind that one time because what could produce this dramatic change? And it was guanfacine. So we then went on to work with it, and it was approved. And it works, by the way, by alpha 2A inhibiting cyclic AMP, calcium opening of those potassium channels. So it's keeping that stress response under control and strengthening connections. And the FDA approved it for treating ADHD, which is a prefrontal disorder, back in 2009, but it's used off-label in a large number of prefrontal disorders, especially stress-related disorders, not surprisingly, and now being tested in delirium to re-coalesce connectivity, and with long COVID. So hopefully it will really help a lot of people.
And so potentially too for, mild cognitive impairment or early Alzheimer's.
I think it would need to be much earlier than that. By the time you get to mild cognitive impairment, you already have cells dying in entorhinal hippocampus. So I think it would have to start very early, as before the calcium has already done its damage. So I've been taking it now for 10 years.
Oh,
So
really?
we'll see what happens to me. I better not get Alzheimer's, right?
You seem to be keeping on task.
Yeah, I notice in particular, I'm always thinking that I must remember to remember. That ability seems to be heightened by guanfacine a lot. Yeah.
Now, one thing that I think might be worth talking about is, so there's some drugs that affect the NMDA receptor, ketamine, which is increasingly used now, I think, for depression, maybe PTSD even, and it will reduce calcium influx through the NMDA receptor. Also, though, it can cause like hallucinations and kind of almost like psychosis, schizophrenia-like state.
Yeah, and profound cognitive impairment. Yes.
How
the hell is this being ever helpful, right? Is what's so intriguing. We have a working hypothesis, and the work of Angela Roberts is very much supporting this, that why ketamine is helpful in depression is because it is reducing neurotransmission in the subgenual cingulate, which is Brodmann's area 25, which is overactive in depression and which activates the entire stress response. So Brodmann's area 25 has extensive connections to amygdala, brainstem, hypothalamus, and it's positioned to activate a stress response, to activate the aversive lens. Dorsolateral prefrontal frontal pole can project down through BA32 to shut off 25, to say, it's okay, we're safe, or to activate it. This room looks really normal, but it's full of virus because look who's in here without masks on, something like that. So
also, Amy, of course, GABAergic neurons, the inhibitory neurons, have NMDA receptors. So,
yes, they also do.
you know, potentially ketamine may be inhibiting an inhibitory neuron, which then leads to increased excitability of the glutamatergic neuron.
So we see it's a dose response. This is not yet published, but that at low doses, really low, yes, you're preferentially engaging the interneurons and stopping them, so you get disinhibition. But then you raise the dose a little bit, and now those pyramidal cells are actually reduced. And this is for the layer 3 ones. It might be the layer 5 are really disinhibited because they don't depend as much on NMDA. From our data suggests layer 5, like the response-related cells, have a large AMPA component. And so when we give ketamine, those cells fire like crazy. So even within the same little subregion, you can have some pyramidal cells disinhibited, but the ones that are doing the representations being reduced. And so we're wondering if Brodmann's area 25 also generates representations using NMDA receptors, but it's representations of pain, of suffering. And so blocking NMDA may stop that representation of pain and be antidepressant. So it's hypothetical, but so far there's data accumulating to support it.
Yeah. Early on, you mentioned that in schizophrenia, there's a pruning of the synapses on the dendrites of the prefrontal layer 3 neurons.
Mm-hmm.
And
that's the work of David Lewis.
And there's also pretty strong evidence that there's pruning of dendrites in severe depression. I think it was maybe first looked at in the hippocampus.
Mm-hmm.
And then there's recently, I think, evidence that ketamine at doses, and the dose is very important, as you pointed out, at doses that have beneficial effects in patients with depression in animal models, those, you know, kind of the same depression relieving dose will actually lead to stimulate the formation of new synapses. So there's pretty good evidence now that with recovery from depression, you can actually reform synapses. So it's not a permanent state. And I don't know if in schizophrenia, it will be interesting to see whether, you know, in the future, you know, really effective drugs, you know, cause formation of new synapses.
Yes. Yeah, no, there was a recent paper using SV2A PET imaging to capture synapses, where you're now able to look at it in a living individual. And they looked at, this is Holmes et al., and they saw that patients with depression and PTSD, their symptoms correlated with synapse loss in dorsolateral prefrontal cortex.
So SV2A is a synaptic vesicle associated protein?
Yes, it's presynaptic, and they've been able to make a PET ligand, and so it's being used now to track synapses in a whole variety of disorders.
I learned that's something I didn't know. That's really, wow. Yeah, that's an important advance, I think.
It's been very revealing for things like this, where I don't think we might have guessed that dorsolateral was so important, because people were focused more on the medial areas. But whether treatment then allows restoration of those synapses, and is there something special about ketamine? There's a lot of research in rodents supporting this idea that you would then be doing it, but and this is not a or, it's probably an and function, that if you quiet BA25 and stop the stress response, you'd also be allowing synapses to reform. There's work by Connor Liston showing that ketamine, the behavior normalizes before the synapses actually return. They were able to actually see that in mouse. So the guess is that it's multi-stage. So you could imagine that the ketamine is first shutting off 25 and allowing you to try to return to normalcy. And then the second stage would be the reforming of the synapses that allows that top-down regulation of emotion.
Yeah. And so you mentioned, all these sources of calcium coming into the neuron, either, coming in from the outside or being released from the endoplasmic reticulum stores. And what about drugs that affect the release of calcium from those internal stores?
Yeah, it would be difficult to get a therapeutic window, I think, because you wouldn't want to interfere with normal function. I should say for our listeners that Mark was one of the most important people for discovering that calcium dysregulation is a key issue for Alzheimer's disease. So your work showing in particular that the presenilins, which are the most common and most aggressive mutations to create early onset Alzheimer's disease. And most of the work in that sphere was all focused on A beta. And you showed this tremendous calcium release from the ER with these mutations. And I think that's why they're so aggressive. People can get Alzheimer's even in their 20s. They can be demented with certain PS1 mutations. Whereas the mutations that duplicate APP, it's a much milder form of disease and much later onset. And these, you also showed, and others, the excess calcium drives A beta formation because it's activating beta secretase. So these are not unrelated.
No.
The calcium, excess calcium drives tau phosphorylation and in multiple ways. And especially when you have enough calcium to activate calpains, calpain 2 in particular, you're cleaving and disinhibiting GSK3 beta, CDK5. So now they can't be stopped. And that leads to the massive hyperphosphorylation of tau, CDK5 when it's cleaved, activating beta secretase. Also activating heat shock proteins to drive autophagic degeneration, which is how neurons die in Alzheimer's disease. The in vitro models often get wrong, thinking it's apoptosis. That's not what happens in Alzheimer's. It's slow autophagic degeneration where neurons eat themselves, and calpain drives that as well. And of course, the synapse loss. So the whole spectrum of pathology that we see in Alzheimer's, the inflammation driven by calcium, and which in turn drives the synapse loss, et cetera. So your work was just central to that. And I'll just add, like Ralph Nixon, also early studies really pointing the way and showing like calpain 2 activated near tangles, that you guys really lit the way for our work. And when we were just trying to study what happens with aging, why is aging such it's the largest risk factor for Alzheimer's, right? Why is it? And why in just these glutamatergic cells involved with higher cognition and memory? And our work led us to calcium, and there you were. So everything that we discovered, it was like you had already seen.
Well, well, that's kind of gratifying. Thanks for that pat on the back. Yeah, so I think, key issue with neurological disorders in general, and particularly neurodegenerative disorders, is why certain neurons seem to be so vulnerable. And these are the kinds of work that is getting at that. And if one, in theory, if one can identify the reasons for selective vulnerability, one might come up with therapies that tap into those mechanisms
Exactly. Early on,
early on.
where
Yeah.
you actually have damage. So like we were comparing primary visual cortex, which is very resistant to Alzheimer's pathology, to prefrontal and entorhinal. And one of the key differences is this calcium magnification, that primary visual cortical neurons didn't have it, prefrontal and entorhinal do. So we think that's a key part of it.
After we did a lot of the work on calcium, which was mainly in the early 90s, actually,
Yeah, a long time.
then we got interested in, and age is a major risk factor for Alzheimer's. Why do some people get it, some don't? We don't know for sure, but there seem to be some risk factors, and one is being sedentary and overindulgent. Couch potato lifestyle. And a third component, I guess, of that is not keeping your mind intellectually challenged throughout your adult life. And so I think I guess I don't know this, but has anyone looked at the prefrontal cortex in terms of getting down to what you're doing, looking at, recording from the layer 3 neurons and so on, with exercise, with intermittent fasting, might I suggest, or just with enriched environments? And I guess a lot of this could be done in the monkeys.
Yeah, we hope they have enriched environments. We work really hard
That's true.
to produce those, and including the cognitive testing as well.
Yeah.
They really enjoy it. There's human data that exercise preferentially improves prefrontal. So I think the fact that prefrontal neurons are just so sensitive to their environment and needing glucose, oxygen, but also we've wondered if lactic acid produced from the muscles that engages a GI coupled receptor that might enhance firing through similar to an alpha 2A receptor inhibiting cyclic AMP.
Or possibly ketones as well.
Yeah, that's a whole area that's very hard for us to
the ketone.
Fun.
Well, yeah, I don't know. There's now ketone ester, essentially beta hydroxybutyrate, which is a ketone that's elevated during fasting, but also during prolonged exercise. And it's been shown, and I had podcasts on this by PET imaging, that if a person is in a ketogenic state, their neurons use mainly the ketones as an energy source, and they quit using glucose. But also, there's work showing that the ketones have signaling functions. They affect gene expression. We showed they actually increase BDNF levels, brain-derived neurotrophic factor. So there actually are some simple ways to look at this. Maybe we could chat about that sometime.
In another
if you're in it, you've got so much going on, but it's another grant. It's another NIH grant.
Yeah, and the long-term health of those brains, and then also other risk factors coming to mind. ApoE4 greatly increases calcium dysregulation. Rhesus monkeys are all ApoE4 homozygotes, so I think that might be why we can see pathology so easily when they get very old.
Interesting.
Isn't that? Yeah. And
So, well, wait a minute, though. That's pretty interesting. So is that true of most non-human primates? Is that known?
I don't know if it's known with other
'Cause now I'm
species.
thinking, well, humans, we have, maybe just one ApoE4 allele or none. We have E3 or E2. Maybe there's some evolutionary advantage to not having E4.
Yeah, well, so for example, and just to bring it back to calcium, ApoE3, calcium, it allows NMDA bursts of calcium coming through, but curtailed. Whereas with ApoE4, it's just continuous, so
Interesting.
larger. And I think, if I'm remembering this paper correctly, also through calcium channels. So the kinetics of it with ApoE3 would be producing a kinetic profile that would be healthier and better for actual signaling of information.
Interesting. Wow. Okay, let's see. There's a couple specific questions I wanted to ask. So you mentioned very early on, you talked a lot about glutamate, GABA, dopamine, norepinephrine, but you did mention acetylcholine, and of course, nicotine activates these, they're called nicotinic receptors, right? What about smokers? And does that have any effect on the prefrontal cortex function?
That's my speculation as to why, especially having caffeine, so the coffee and the cigarette,
Oh,
the caffeine inhibits adenosine, it blocks the adenosine receptors that are inhibiting the cholinergic receptor. So you'd get more acetylcholine release in prefrontal. And then the nicotine, you'd be getting this additional burst. So my fantasy is the reason that's what people do to be able to write and work is because you're maximizing stimulation of those alpha-7 nicotinic receptors. Makes me almost want to vape, but it would have to be a specific
it's
alpha-7.
addictive. It's addictive.
Yes, no, and the addictive is the beta 2, alpha-4 beta 2, for example. So if we could vape a very low affinity alpha-7 nicotinic agonist, I'd be interested.
You could do that in addition to the guanfacine.
That's right. Yeah. So then I'd have a little phasic enhancement. Yeah.
But have there been clinical trials looking at cognitive function with guanfacine?
Yeah. So for example, there was a small study showing that it enhances cognitive remediation therapy in patients with schizophrenia syndrome spectrum. And then there's also, of course, it was approved by the FDA based on rating scales for ADHD, where it's, can you, focus, pay attention, impulse control, things like that. But also there have been imaging studies with working memory.
Okay, Amy, I think we can end here. I'm going to put in the links, there's a description section on YouTube and actually also on Spotify and the other podcasts for it. I'll put links to some of your articles on your one on Pat Goldman-Rakic,
Oh, yes.
and her career, and then a couple of your review articles related to chronic stress and to the kind of things you've talked about here in this podcast. So people, I also put in links to a couple of your scientific lectures.
Oh, I've also made some YouTube videos that are on the Yale Medical School channel about prefrontal and mental disorders, the neurobiology of burnout, things like that. So I can give you those links as well.
Yeah, send those to me by email.
This has been just a delight, Mark.
Okay, Amy, thanks again. Bye.